Okay, and now it's time to move to the last session of this webinar, evaluation of drug-induced liver toxicity in a 3D HepaRG-based microphysiological human liver model. So we welcome for this talk Dr. Knut Rennert from Dynamic 42. Knut is co-founder and CEO of Dynamic 42, a biotech company specialized in human in vitro organ on-a-chip models. Knut has been working with on-chip cell culture applications since 2020, since 2010, excuse me, and as part of his work at the University Hospital Jena at Germany, he completed his doctorate degree in 2005 in basic research of cancer biology and worked during his postdoc in the field of atherosclerosis. Here he first-hand experienced the need of physiological human blood vessel and organ models that properly represent the situation in vivo. Thanks, Knut, for being with us today, and it's yours.
Okay, can you see my screen?
Yes.
You can do. Okay, then Pamela, thank you for the kind introduction and also for the invitation that I can take part and present our data in the HepaRG webinar. Briefly regarding our company, the Dynamic 42, we are CRO, we are a contract research organization that was founded in spring 2018. And we are situated in Jena in the middle of Germany, and we are working with BioPredict for more than now 10 years. So also earlier to our founding the company, we already worked with HepaRG in a research group of Alexander Mosi's lab here in Jena. And as already mentioned, we are developing and commercializing organ-on-a-chip platforms, also known as microphysiological systems, and they improve then the data quality already in the preclinical phase. And here I will now present you a case study that we have worked on in the last years regarding the evaluation of drug-induced liver toxicity in a 3D HepaRG-based microphysiological human liver model. First, drug-induced liver injury is the main cause for drug withdrawal in the drug development process. On the right in the circle, you see areas which are, or what are the reasons for withdrawal by drug-induced liver injury. And you see about 1/3 is dealing or has toxicity problems, let's say, in the hepatocytes in the liver. And the major health threat in drug-induced liver injury is also a major health threat for the human patients due for difficult predictability and also the clinical diagnosis. Drug-induced liver injury will be divided into different areas in the intrinsic DILI and the idiosyncratic. The intrinsic has characteristics that it's predictable, it's dose dependent, and it's mostly acute toxicity. An example for this is acetaminophen, also known as paracetamol. And on the other side, you have the idiosyncratic DILI. It's non-predictable. There you have no dose dependency and often also variable latency time. And here, antibiotics like amoxicillin or trofloxacin are situated, for instance. So to uncover occurrence of drug-induced liver injury before human clinical trials and preclinical investigations were performed. You know this already in 3D culture, 2D culture, rodent models, and so on. All these models have their advantages and disadvantages. So we have the 2D, for instance, 2D cell culture, which is good for reproducibility and also for controllability, but the physiological relevance is really low. But on the other hand, you also have animal models that has high physiological relevance and complexity. However, there are a lot, as also Leo mentioned, interspecies variances between the animals and the humans, and that's why you also have low predictability of, for instance, liver tox in humans. And one alternative that bring the advantages of both systems, let's say, together, is the microfluidic organ on a chip. And I will present now some data of our case study. Okay, the dynamic 42 liver model is inspired by the human liver anatomy. So we focused here on the smallest functional unit of the liver, which is the liver sinusoid, and their cellular arrangement. For this, we established a chip platform. We have a cavity that is separated by porous membrane, which gives you an upper and a lower chamber or channel. And in the upper channel, you can integrate the vascularity consisting of liver sinusoidal endothelial cells, as well as a surrogate for Kupffer cells, in this case here for monocyte-derived macrophages. Beneath the membrane, we are able then to integrate differentiated HepaRG, hepatocytes. You can do it in a gel, but you can also do it only by coating. And there you have also the structure of these liver sinusoid in the chip. And one advantage of our model is that you can perfuse the vascular site with media, so thereby mimicking the sinusoidal bloodstream and and bring it more in a physiological range, let's say. Here, I slightly show the liver model assembly. So you start with a chip, you sterilize it, bring in a coating if necessary for the liver model. You integrate, for instance, extracellular matrix like collagens. Afterwards, you seed in the liver sinusoidal endothelial cells in the top channel, wait till they grow to confluency. Then you integrate the monocyte-derived macrophages in the vascular part. And after a few hours, days, you then integrate differentiated HepaRG into the bottom chamber, turn it upside down that they can adhere to the membrane, and then culture them again for 1-2 days, and then start perfusion that the system can adapt to the flow, to the blood flow. You can do a pre-perfusion, and then you are able within 1 week to start the drug perfusion or the drug experiments. As mentioned, we focused here in this case study on trovafloxacin and levofloxacin. Trovafloxacin is an antibiotic that was approved in 1997. 2 years later, it was withdrawn from the market because there were over 140 severe adverse hepatic effects, and 10% out of it showed acute hepatic toxicity. That's why it was withdrawn from the market. In the next years, the toxicity mechanism was analyzed. It is a immune-mediated cellular ATP depletion. And in humans, the plasma concentration is roughly about 10 micromolar. And that's why we also choose a 10-fold lower concentration. We use the plasma concentration and doubled Cmax for our experiments. To compare it with a control substance or compound, we use levofloxacin. It's an analog of trovafloxacin, but it's known that it's non-hepatotoxic, not at this concentration. And here we have comparable plasma concentrations of compared to trovafloxacin, and so we can also take or apply the same doses like for trovafloxacin. And these antibiotics or the compounds were then daily applied to the system via the perfusion, and we cultured the liver on chip for up to 7 days. In the end, we performed several analyses, starting with fluorescence microscopy. So we checked for morphological analysis, marker expression, also for live cell staining. Then Tim also performed automated quantification. So he made the IF, or he stained the nuclei by DAPI and counted them, the occurrence in the image that he has recorded, and could then check for cytotoxicity. And we also sampled the effluent, medium, and check here for damage parameters like lactate dehydrogenase, LDH, and also for ALT, and also for potential immune cell activation by checking for pro-inflammatory cytokines. So the first analysis that we did was regarding the marker expression and also the morphology of our liver chip. We started with the vascular side. So here we had a look on the marker expression of the liver sinusoidal endothelial cells. So CD32B is one of these markers. And we have a look, we analyzed how increasing concentrations of trovafloxacin have an effect on the expression of this marker. And also as a control, levofloxacin was also analyzed. And what you see that you get a reduction in signal intensity when you look on the immunofluorescence stainings by increasing concentrations of trovafloxacin, which is not visible at high concentrations of levofloxacin. The quantification or the bar regarding the fluorescence intensity analysis is also shown on the right. You see it's not significant, however, you see a trend. The second marker that we analyzed was CD206, also known as mannose receptor, and macrophages as well as the liver sinusoidal endothelial cells express it, but they can be easily separated in the analysis. And what you see with increasing concentrations of trovafloxacin, you see a decrease in signal intensity for the macrophages, and you also see it. It's not here visible, but it was also analyzed by Tim, that also the liver sinusoidal endothelial cells show significant reduction of CD206 at Cmax of trovafloxacin. Taken together, one could say, okay, cytotoxicity and decreased marker expression occurred after the application of trovafloxacin concentration starting at 10 micromolar. And levofloxacin, there were no toxic effect visible. In the next step, we analyzed the hepatic compartment, so the HepaRG. Here we compared marker expression of the hepatocytes and looked also for the influence of increasing concentrations of trovafloxacin, starting with the asialoglycoprotein receptor 1. You could see with increasing concentrations of trovafloxacin, a decrease in signal intensity. This was, could you also see that it got significant differences here. This was also checked for CYP3A4 enzyme, the metabolizing enzyme in the hepatocytes. Here you couldn't find, we couldn't find a significant difference, but you see that the hepatocytes are really harmed at high concentrations of trovafloxacin. And we also checked for alpha-glutathione S transferase, and you can also see here, the layer is harmed, and you also see a significant decrease in fluorescence intensity. So you see hepatotoxicity occurs here in the hepatic compartment or in the HepaRG when you use concentrations of trovafloxacin that were 10 micromolar or higher, and levofloxacin didn't show any hepatic injury, but a slight decrease in the marker expression. Coming now to the analysis of the cellular viability. On the left side, you see bright field images where you see that at plasma concentrations of trovafloxacin, 10 micromolar, you get some holes and some areas where cells are not attached anymore. This is for levofloxacin, it couldn't be shown. Then an analysis was done regarding the DAPI nuclei staining So it was measured by, or it was recorded by fluorescence microscopy and then quantified. So nuclei were counted per field in these images. And what do you see is that you have a strong decrease of hepatic cells and also vascular cells with high concentrations or with increasing concentrations of trovafloxacin, but this is not visible for the levofloxacin. Okay, so you see trovafloxacin induced the loss of the nuclei in the vascular as well as in the hepatic cell layers. Another step was then to measure damage parameters from the effluent. So you are able to sample at each time point you like from the system. Here we checked, so we sampled from the vascular side and investigated how ALT release or LDH release will be affected by increasing concentration of the different compounds. And for ALT, you see after 48 hours, there is a trend that trovafloxacin will increase the ALT release. This is more prominent and then also significant after 72 hours compared to the control as well as to the levofloxacin, the non-toxic analog, which did not increase the ALT. And then this is more specific for the hepatocytes, and we also checked for the LDH. So this is more cell damage marker for all the whole liver model, let's say. And here you also can see that concentrations of 20 micromolar trovafloxacin significantly increase the release of the LDH already after 48 hours and also after 72 hours. So, and levofloxacin is still comparable to the control. It did not enhance the LDH release. Okay. As already mentioned, we also checked for cytokine profiles. So as we have immune cells or macrophages in the system on the vascular side, we also checked if cytokines will be expressed and released in the liver chip. And herefore, we checked for different pro-inflammatory cytokines. The most prominent effect we saw for the IL-1 beta after 48 hours and the highest concentration of trovafloxacin compared to the control or to the analog. You see there's a significant increase. Also, we see an induction of the release or the expression and release, for TNF-alpha, IL-18 and IL-8 in our liver-on-a chip. And what else do we see? That IL-6 was reduced at the highest concentration of trovafloxacin already after 48 hours and also after 72 hours. And that levofloxacin did not significantly alter the cytokine profile. In a further step, we then moved on to live cell imaging to get also a little bit a view how the toxicity mechanism could look in or could be in our liver-on-chip model. At first, we checked for glutathione level of the hepatocytes. We stained here the liver-on-chip model with monochlorobiman. This gives you, in this case, blue signal. And you see with increasing concentration of trovafloxacin, these signal decreases, and compared to levofloxacin, where it's more or less the same compared to the control. This was also quantified and analyzed by a software fluorescence intensity. You see that you have significantly decrease of glutathione level in trovafloxacin treated liver-on-chip models. Further on, we checked for mitochondrial activity and integrity, as you already saw in a few slides before, that the cell layer is not that consistent anymore with increasing concentrations of trovafloxacin. And here you also see that the signal intensity decreases with increasing concentration of trovafloxacin. Here you see it's not significantly altered, but there's already a trend, sorry. In the last step, we looked for, or we analyzed for the production or the release of reactive oxygen species. As we know, this could also be a mechanism that could lead to hepatotoxicity. And what do you see here is in control as well under treatment with levofloxacin, there is no increase in ROS production. However, when you treat the liver model with trovafloxacin, 10 micromolar and 20 micromolar, you see a significantly strong increase in ROS production. So this means trovafloxacin induced the decrease of the glutathione and an increase in the ROS release, but levofloxacin maintains the redox status of our liver model. In the end, we can say that these liver-on-chip model with HepaRG was suitable to detect the trovafloxacin-induced drug-induced liver injury, and also that it indicated a non-toxic profile for the non-toxic analog levofloxacin at human therapeutic concentrations. It also turns out that in contrast to mice models, the toxic effect of the trovafloxacin was shown without further treatment like LPS or TNF-alpha. And I think you could also see that it has a high potential, this model, to detect adverse side effects that were not demonstrated then also in 2D cultures or in animal models. For the future, we are currently screening for more DILI drugs to validate the model. We are already in the development or establishing phase of multi-organ models. For instance, we can combine intestine model and liver model for investigations regarding toxicity. Also, lung and liver is also established. And we are currently also working on the development of medium throughput platform, as we know that our chip, it's only currently in a low throughput form, but for drug screening and pharma companies, they need much higher throughput. So here we are also working on further developments. So I would like to thank you for your attention. And I would like to thank mainly Tim Carden, because he's an outstanding scientist and he has done really great job, and he is always doing great job. So all the work that I've presented here was done by him. And I also like to thank the team. Without the team, it was not possible to perform this study. I also like to thank Dr. Alexander Moses' lab for support and for mentoring also Tim. And I would like also to thank Biopredict, Pamela, as well as Christoph, for the possibility that we could attend and present our data here in the HepaRG webinar. So here at the end, there are the references that were used or that the data that I have shown. Thank you very much. And I'm open for your questions.
Thank you, Knut, for this nice presentation. The first question, how many chips are on one plate and how high throughput is the system?
So it's currently in a microscopic slide format. So you have two models that you can have on one chip, but we are currently working on a platform that we put it more in a SBS format, like a 96-word plate format. However, I think the next step would be 8 to 12 models, let's say, that can be established in parallel. So it's really, it's low throughput, but one scientist can handle between 10 to 12 chips in parallel. So this means you have 20 to 24 samples, let's say, that one scientist can handle. So it's not for early drug screening, it's more when you have special questions already. So mechanistic mode of action questions, something like this.
Okay, thank you, Alan, for your questions. The first one, how long did it take to collect this entire data set?
To take the entire data set. So you mean how long the case study took, or is it more? So 1 week you need for establishing or setting up the model, and then you have 1 week to get the effluent, and yeah, you have the endpoint analysis. So it depends a little bit what you would do, but I think from start till the end, start with sawing cells or bring in the cells into the chip, and analysis, it takes for one experiment, let's say 3 weeks, 3 to 4 weeks.
Okay. And how many chips were you able to maintain in cell culture at steady state?
So, more, what does, can it more be?
Alan, can you elaborate more your question? To maintain cell culture at steady state for how many days you can maintain the cells in this kind of system?
So yeah, got it. So the functionality of the liver model is 14 days. So it's stable till 14 days.
Okay, and what kind of success ratio can you expect from using these chips until the endpoint of 14 days?
In which, what could you define it more? So
yes, I will ask Alan. Alan, maybe you can elaborate more on your questions. You
hello.
can speak now, Alan.
Hello.
Yes, hello.
You can hear me?
Yes, I can do.
Okay, great. Sorry, thanks. It's very nice work because I think this is the first kind of comparative study which I saw. So I was more curious about this. So I was wondering, like, do you have any kind of failures during your culture, like let's say the perfusion failed, or like there's some inherent bias, like the cells deloaded for some other reason? Like, can you culture around 10 chips continuously for 14 days with a 100% success ratio from your experience?
Okay, in this direction. So I think the success rate is about 80%, 80 to 90.
Okay.
So we usually also have some backup chips in it.
Okay. Okay. But your scientist was able to maintain at least 10 chips continuously for the entire duration of your experiments. Like
so
that's
in this study, not, because this study was not only, but here we looked for toxicity after 3 to 7 days. But we, upfront to his study, we are able to keep the liver model functional for up to 14 days. So, but for this case study, it was also, did not make sense to go for the 14 days because we saw toxicity effects already after 3, 5, 7 days.
okay. Thank you. So just the last question is that have you thought about extracting the tissue after your culture for transcriptomic based analysis? Is it possible in the device, and have you tried it already?
It's possible. So we have different chip designs. It depends a little bit which design you take or you use, and because that you get enough cell material out of it. But in principle, it's possible to isolate RNA from it and to also then perform PCR and sequencing, something like this. It's possible. You can easily open the chip at the end as an endpoint and recover the tissue out of the box or out of the chips. Sorry.
just to get an idea, how many cells are there in each layer for hepatocytes and the endothelial cells?
So for the hepatocytes, it's depending on the design, something between 200 to 400,000 cells. And on the LSAC vascular side, it's roughly also the same. So like 350,000, something like this.
Thank you so much.
Yeah, you're welcome.
Thank you, Alan. Knut, did you think about the use of co-cultured HepaRG for more physiological relevance of the model? So in the layer of hepatocyte, no, co-culture of, maybe the question is about co-culturing HepaRG cells with other than the vascular layer, other cell types, like in the layer of hepatocytes.
Yeah, so we have stellate cells, for instance.
Some stellate cells.
Mm-hmm. So we already thought about it. We also had experiments, for instance, with the LX2 cell line. However, we had a lot of problems because this LX2 overgrows the culture, so that within, I think, 3 or 4 days. So it was very, very fast. And regarding the integration of stellate cells, we already did this. However, so far we did not have questions from pharma companies. They were accepting, or they thought the triple co-culture model out of LSECs, macrophages, and the hepatocytes are suitable enough. So, or the that's why. But it's in principle it's possible. So there's the stellate cells. We verified their presence in the layer by PDGF receptor beta, I think. Yeah.
Okay. And what type of media did you use in your system for the co-culture of HepaRG and vascular cells?
As these channels are individually perfusable, these cells, or we are able to stick to the cell type specific media. So for the vascular side, EC media with supplements for the macrophages, that they are still happy for 14 days. And on the hepatic side, it's a cell type specific media for
the regular medium of HepaRG cells. Okay. And have you tried this with primary human hepatocytes?
Yes, we already did, and it's also working, but there are also the problem that Leo also mentioned, I think, in the beginning. The differences between the batches and the donors is really high. And yeah, some effects from one donor are not really, not always, let's say, reproducible not reproducible compared to the other donor. And so there are different impacts, let's say.
Okay. Not very technical question. What is the flow speed did you use?
So currently we are running our systems between 25 to 50 microliter per minute.
Okay. And can we get the ratio of HepaRG, MDM, LS, stellate, endothelial cells used? And have you done any drug metabolic conversion studies using liver models on other tumor models? And how important is using stellate cells with this model? This part of the question we already answered. Yes, it may be very interesting. So the ratio of different cells, and have you done any drug metabolic conversion studies using liver models on other tumor models?
Okay. Regarding the first questions, I know that Tim really orientated on the physiological ratio, and I think that this was also then integrated into the chip. But and the second question regarding
drug metabolic conversion studies using liver models on other tumor models. So I don't know if they want to say that hepatocytes, let's clarify that HepaRG cells is not considered, HepaRG cell model is not considered as tumor model. So did you try with other drug metabolic conversion studies with other drugs on other types of cells?
So in the liver model, we also make a validation study currently with Tolka, entacapone, for instance, ximelagatran, dapigatran, and also troglitazone and pioglitazone. And regarding other tumor models, so we are developing currently a pancreatic ductal adenoma carcinoma model, but so far there were no drug testing in it. So we are currently in the development phase.
Okay, and as liver model, you tried HepaRG cells and primary human hepatocytes,
Yes.
but not other. Okay.
No,
Okay.
no HepG2. No. Okay.
Okay. And how do you explain the reduction of IL-6 after trovafloxacin treatment? How, whereas IL-1 increases, IL-6 decreases. So how can you explain the reduction of this IL-6?
Okay, so the IL-1 beta, I think it's a short-term increase. So it's important to have a strong response very fast to recruit, I think, further immune cells. And for the IL-6, I think it's a little bit because it's produced from not only by macrophages, but also by further cell types. It could be that one explanation could be that, as you have also seen in the analysis for the hepatic marker expression, that the hepatic cells were really impaired. So there were lower cell amount. It could be that the IL-6 reduction is a result of the reduction of cells because they are killed or damaged by the trovafloxacin.
Okay.
Could be one explanation.
Yeah. And did you verify the suitability of the liver model for drug-induced liver injury with any other compound?
Yes. So we are currently doing this. We are validating the model with drugs like that. We also know we have a toxic and a non-toxic analog. And we have here 3 pairs currently: tolcapone, entacapone, ximelagatran, dapigatran, yeah, and troglitazone and pioglitazone, sorry.
Okay.
Great.
And the last question, I think, small molecules are often hydrophobic and tend to stick to the ship material. Did you analyze the absorption of the compounds to your ship material?
And yes, we do this always upfront to the testing in the liver model, or in the liver, or in the organ model, let's say. And we checked different compounds also for the absorption in the free cell system. So, for instance, for propiconazole, it's a very hydrophobic one. Small molecule has a log P of 3.7. And here we saw that only 80 to 85% of the compound, not only 80 to 85% of the compound are still in the medium and are accessible to the cells. So this is also an advantage versus other platforms where, for instance, PDMS tend to absorb a lot of hydrophobic molecules. Yes.
Okay.
And I like to mention because in the first 2 talks there was spoken about a lot of spheroids. We also further developed our chip design, so we are currently also we have a 3-chamber model, so it's not only 2 channels for vascularity and, let's say, for the tissue. In the 3-channel chip, we are able to integrate a vascular side, and then we also have integrated microcavity membrane, where you can in each cavity, and not in each cavity, for each sample, let's say, chip, you can culture up to 25 spheroids in parallel. So maybe this is also some interesting feature or chip that could be useful also for further investigation by Leo altogether.
It's very interesting.
Okay.
Thank you, Knut.
Yeah,
so that was everything. No more questions. Before we wrap up today's webinar, I want to thank you all for being a part of this event. I also want to extend a big thank you to our fantastic speakers, Dr. Leo van Glusman, Dr. Guillaume Tournière, and Dr. Knut Rennert for sharing their expertise and the insights with us. More webinars will be held in the near future, so stay tuned. Keep an eye on your inbox for updates on upcoming events. Until next time, goodbye for now.
Goodbye.